Greetings everybody. So today we are going to discuss about a new topic which is the fungi. So first we are going to discuss about its general characteristics. What are fungi? Fungi are those organisms which are unique to both plants and animals such that they have some characteristics of plants, some of animals. It comes to their plant-like characteristics, they have plant-like characteristics such that they possess cell wall. They do not have centrioles, as well as they are non-motile. And in plant animal like characteristics, they do not have plastids, as well as they um are heterotrophic nutrition. They do not have plastids, as well as they have chitinous cell wall, as well as they have reserve food as glycogen and oil rather than starch. And these are some characteristics which are both unique to both characteristics. Moreover, when it comes to some other unique characteristics of fungi basically uh, are absorptive heterotrophs. These are absorptive heterotrophs, which means they do depend on other organisms for their food, but they obtain their food through the method of absorption, such that um, those heterotrophs which are absorptive, they tend to absorb their food by the help of special structures which are known as rhizopods. Uh, uh, Rhizopods, so which they tend to absorb that structure. Now, they have a body which is known as mycelium. Body which is known as mycelium. They all are multicellular except one phylum, which is unicellular. Which includes Deuteromycota. We are going to discuss that. And mycelium, as well as their body is a simple body mycelium, which is having several thin thread-like structures, which are known as the thin thread-like structures, which are known as the hypha. Sorry, hypha is for singular and hyphae plural. They have thin thread-like structures, hyphae, and these structures, hyphae, are of different types. First and foremost, when it comes to mycelium, their structure mycelium is divided into two types, which include septate and aseptate, as given in a diagram as well. Moreover, fungi have a unique characteristic that they have, instead of performing mitosis, they have nuclear mitosis, such that um, they have no degradation of nuclear membrane during mitosis, and the formation of spindle fibers during the metaphase, and their release and during the metaphase occurs within the nuclear membrane within the nucleus. So this is a special type of mitosis occurring within them, which is known as nuclear mitosis. This was a one unique characteristic, and second unique characteristic was that when the genetic constitution was checked out, when the genetic constitution was uh, checked, that what is their genetic constitution, it was found that they have genetic constitution of their cell as well as their mitochondria, neither similar to plants nor similar to animals, due to which they were placed in a separate kingdom which was known as fungi. And these are also known as mycota. This kingdom is also known as mycota, and person who studies mycotes or fungi is known as mycologist, and the study is known as mycology. And apart from that, absorptive heterotrophs are there. And when it comes to the mycelium, it is a simple body of fungi, and here it is divided into two types. First include a septate and aseptate. They have two types of bodies, such that either they are having a cenocytic body, which is known as cenocytic hypha. Cenocytic hyphae is there, which is known as cenocytic hyphae, which is known as aseptate hyphae. And that aseptate hyphae is you know having no cross walls between them. Cenocytic hyphae, aseptate hyphae. Example of this cenocytic hyphae involves um, rhizopus. Rhizopus is an example of this. Rhizopus. And when it comes to another type, we are having septate hyphae. Septate hyphae. And these septate hyphae basically are having cross walls between them. And as these having cross walls between them, these cross walls are having several perforations in between them, so that food material can be transferred from one cell to another. And example of this includes penicillium, from which the first vaccine was discovered by Edward Jenner. Penicillium, as well as the septate hyphae have cross walls, and example of this includes penicillium, right? And where there are four major groups in which the fungi are divided. Number one is zygomycota, also known as conjugating fungi. Number two includes basidiomycota, also known as club fungi. Escomycota, known as the sac fungi. And no, basidiomycota is a sac fungi, escomycota is a club fungi, right? And then deuteromycota, fungi imperfecti. Now, when it comes to out of these four major groups, when it comes to the cenocytic hyphae is only found in zygomycota. Zygomycota. These are known as conjugating mycota. The reason behind it is that they perform conjugation. Now we have studied what is conjugation. It is a process in which two prokaryotes, I mean not necessarily two cells, uh, you know exchange their genetic material between them, which is known as conjugation, and it is a sexual reproductive process, right? After that, when it comes to septate hyphae is found in basidiomycota and escomycota. Escomycota. Moreover, when it comes to some other structure, we are also having septate. We are also having escomycota, basidiomycota, and the mycelium are also classified on the basis of number of nuclei, such that if we are having mycelium which is having only one nucleus, then that would be considered as monokaryotic, and that would be considered as the primary mycelium. Primary mycelium. If it is having two nuclei, dikaryotic, then it would be considered as secondary mycelium. Secondary mycelium, right? And the hyphae can be divided into several parts. Number one includes a reproductive part, that there can be reproductive hyphae, and there can be vegetative hyphae or absorptive hyphae. Reproductive hyphae and vegetative hyphae. When it comes to reproductive hyphae includes is a vertical erectly growing hyphae, which includes sporangiospore and conidiospore. Sporangiospore and conidiospore, whereas the absorptive hyphae are of two types. Absorptive hyphae involves those hyphae which is found in those organisms which are saprophytic, saprotrophic, uh, which absorb dead organic matter from the soil, which includes uh, you know examples of mushrooms in them. And these hyphae found in these organisms are known as rhizopods, known as rhizopods, right? And apart from that, those hyphae which are found in parasitic mode of life are basically known as the hostoria, right? After that, sporangiospore. Rhizopods are found in absorptive mode of nutrition, but hostoria is also found in absorptive mode of nutrition, but in parasitic mode of nutrition. Moreover, when it comes to the structure of sporangiospore is a type of structure, asexual reproductive structure, which contains, it is a modified hyphae, erectly growing hyphae. It is asexual reproductively growing hyphae, which contains, at the tip of it, there is a sac-like structure which is known as sporangia, and that sporangia contains and is a site of production of spores. These spores are asexual uh, agents, asexual cells. These spores are asexual cells which are motile, while when it comes to which burst out when the sporangia bursts out, they grow into a new fungi. When it comes to conidiospore are cells. These basically are having a similar structure as sporangiospore. Likewise, if you're supposed to draw the structure, conidiospore, this would be the complete conidiospore, and over the tip of it there would be a sac-like structure known as conidia. Conidium for singular, conidia for plural. Conidia, right? Conidia would be there, and within that conidia there is production of several conidiospores, and these conidiospores again burst out in order to form in order to grow into separate organisms. Now, examples of those which contains sporangiospores are Rhizopus. Rhizopus has sporangiospore development, and Penicillium has development of conidiospores. After that, these are the two modes of asexual reproduction. Another mode of asexual reproduction is generally involved, which is known as budding. Budding is a process in which nuclear mitosis occur within the cell of mycelium. Nuclear mitosis occur in the hyphae cell, sorry, and as a nuclear mitosis occur, that nuclear mitosis leads to the formation of two nuclei, and once these two nuclei are formed, the other nuclei one of the nucleus causes one of the nuclei causes to uh, per, protrude out as an outgrowth of of the cell, protrude out as an outgrowth of the cell, and gradually is protruded out and taken out as an outgrowth, and grows ultimately into an other mycelium. Moreover, there's another process occurring which is known as fragmentation, and fragmentation is a process which occurs when the mycelium when a part of mycelium is broken, a part of mycelium is taken out and grows into a part of mycelium is broken a part of mycelium is broken and taken out into part of mycelium is broken, and that individually grows into a new mycelium, which is known as fragmentation. And budding occurs in my hyphae, right? So budding occurs in hyphae and fragmentation in the mycelium, right? And after that, when it comes to some other part, these are the asexual reproductive processes. And when it comes to sexual reproductive processes, um, before discussing sexual reproductive processes, let us discuss the evolution point of view, that how algae are believed to evolve. Algae are believed to evolve from organisms, from sponges. They have same evolutionary linkages, just like that of sponges with conoflagellates, such that they form monophyletic linkage. They form monophyletic linkage, and that monophyletic linkage is believed to have occurred. They are believed to have evolved around 570 million years ago. 570 million years ago, and monophyletic linkage, which is believed to have occurred which is believed to have occurred 570 million years ago. They form monophyletic linkages from um, sponges like ancestors with red algae, such that red algae and is believed to be the ancestral root of fungi, such that the evidence behind it is that red algae also do not have any sort of flagella, as well as red algae are also having a characteristic of absorption in some cases. So in this case, fungi can it can be the possibility, but still it is only believed that the evolution is occurred from red algae, because both of these are non-motile structure. Both red algae as well as fungi do not have an both of these are uh, non-motile structures. Both do not have any sort of flagella, so it is possibility that fungi are evolved from red algae. Why not euglena? Euglena can could have been the possibility for the evolution of fungi, but reason behind it is that euglena is having flagella. And there can be other organisms, but other organisms are also having flagella at some stage of their life cycle, but red algae are not having so. So these protoctist red algae are supposed to be the part from where the fungi are evolved. And one more question arises, that why are fungi not placed in protoctist? If they were that much unique, then they should have been placed in protoctist. But again, if we were supposed to place fungi in the protoctist, then it would be simply clarified that protoctists are evolved from prokaryotes, while the monophyletic linkage of fungi does not show a correlation with prokaryotes. Rather, it shows correlation of evolution with other eukaryotes. So it cannot be concluded that fungi are evolved from prokaryotes, right? In order to since they are not evolved from prokaryotes, we cannot place them in protoctista. Rather, we place them in fungi and consider them as a separate kingdom which evolved from the protoctist and form monophyletic linkages with the protoctist, particularly the red algae. Moreover, uh, one more thing, back to the normal point, which is sexual reproduction. Now, sexual reproduction occurs in a manner which is known as conjugation. Either occurs through conjugation. Conjugation process already told that in which if we are having two cells which are having two different genetic material and both exchange their genetic material, then they tend to uh, perform conjugation process between them, as it occurs in prokaryotes. And another process which occurs is known as plasmogamy. In terms of sexual reproduction, all fungi perform sexual reproduction except Deuteromycota, because they are unicellular. Example of this include Saccharomyces. Moreover, Saccharomyces and Syncy- Syncytium. Saccharomyces and Syncytium and Saccharomyces. Syncytium and Saccharomyces. Syncytium, Syncytium and Saccharomyces. Syncytium and Saccharomyces. Both of these are unicellular fungi involved in Deuteromycota, also known as fungi imperfecti, and both of these do not have mycelium. So in these are lower fungi which do not have mycelium, Deuteromycota, and these are lower fungi which do not have mycelium, lower fungi which do not have mycelium, lower fungi don't have mycelium, right? So these are some characteristics, and apart from that, a conjugation occurs in the multicellular ones, as well as there's another process occurring which is known as karyogamy. In all of the sexually reproductive fungi, uh, two processes are very common: meiosis, number one, and number two is the fusion of haploid gametes. Fusion of haploid gametes. Of course, we are not having formation of gametes, but in that case, we would be having fusion of haploid nuclei. Fusion of haploid nuclei. And when this fusion of haploid nuclei will occur, that would be considered as karyogamy. Now, how this process will occur? Let's discuss. So here, the process will occur in such a manner that first, the two haploid cells, which are having the two haploid nuclei, will tend to, perform a process which is known as plasmogamy. Plasmogamy, which means their cytoplasm will be mixed. And once the cytoplasmogamy occurs, after that, there will be process of karyogamy occurring. Karyogamy will occur, and that karyogamy means fusion of the two haploid nuclei, due to which there would be formation of a diploid nuclei, and that process which would be karyogamy. But in some groups of fungi, for example, basidio mycotes, basidiomycotes, and acid ascomycotes, there is no formation of karyogamy. Rather, the two haploid nuclei coexist independently within the cell, due to which these two nuclei existing independently within the cell remain functionable within the cell for the rest of the life of the fungus, and they remain within the cell until they are either either the cell is dead, or they're reproduced, they perform reproduction once again. So at that case, these fungi, these what do we call it, these hypha, because this process would be occurring in hypha, because hypha is responsible for the asexual or whether the sexual reproduction. So in this case, this process of reproduction occurring in hypha, so we will be considering that cell as hypha. So this hypha, these hyphae, or this hypha, because it is a singular, this hypha which is having this hypha which is having two nuclei of two different genetic constitutions, of two different genetic constitutions are known as this hypha which is having a nuclei, this is having two nuclei of two different genetic constitutions together, are known as the heterokaryotic or dikaryotic hypha or cell. Heterokaryotic and dikaryotic hypha or cell. So this is a part about reproduction. Now coming to the other part, which is about their habitat. How do they live? So generally talking, they grow in moist habitats best, but they can also survive in dry habitat readily. Um, if there is dry habitat, so they can grow in their reading stage, in their resting stage. They can grow in a dry habitat in their resting stage, as well as when by the formation of resistant spores. Moreover, they can survive a pH of 2 to 9, pH of 2 to 9, survive a pH of 2 to 9. Moreover, they can survive high temperature as well as high osmotic pressure, high temperature and high osmotic pressure. Osmotic pressure here refers to condition in which the pressure of the fluid is more, such as salt concentration, high salt concentration has high osmotic pressure, high sugar concentration has high amount of osmotic pressure. In that case, they can also survive in such condition. Example of this includes jelly or jam, in which they can survive, jelly, jam, right? And these conditions allow them to survive on terrestrial areas. And apart from that, they contain around 110 million species, if I'm not wrong. 100,000, 10,000, or sorry, 1 million species, right? Not 10 million, 1 million species. They contain almost 1 million species, right? And other MCQs point I like to highlight over here is about uh it is about its mode of nutrition. Mode of nutrition is divided into several types, such as saprotrophic. And saprotrophic mode of nutrition, the hypha which would be modified would be known as rhizopod. Sorry, it's not rhizopod, it's rhizoid. Rhizoid would be the hypha. And one more thing, that here their separate mode of nutrition would be in such a manner that they will tend to release, usually they feed on dead matter. But if they have a requirement of digesting the food, at that moment they will tend to release their digestive enzyme outside their cell. And at that moment, when the digestive material, enzymes will be left outside the cell, they will tend to react and break that down into the substrates, break the substrates down into the products, which would be absorbed by the rhizoids into their body, mycelium. Right, so this is how the saprotrophic nutrition will occur. After that, when it comes to the parasitic nutrition will occur in such a manner that in parasitic nutrition, they will absorb the food material from their host cells, either intracellularly or intracellularly. These are endoparasites, right? Endoparasites. And here that can occur either intercellularly or intracellularly. And parasitic mode of nutrition will occur through formation of hostoria. After that, after formation of hostoria, after formation of hostoria, I mean, these are already structures present. They can be obligative parasites and facultative parasites. Both of these would be having the hostoria, right? And when it comes to obligative parasites are those obligate parasites. Sorry, obligate parasites. These are parasites which which obtain their food from the host. They can live on their host, but they cannot survive other than other favorable conditions other than their host. Such that if they are able to survive on their particular host, they cannot survive in an artificial culture media which provides the same favorable conditions in order to survive in the host body. So in that case, these would be considered as obligate parasites, and these obligate parasites include examples of mildews and rust species. Other example is facultative parasites. Facultative parasites are those which are having, uh, these can live in on the host, that they can survive on the host, inside the host, as well as they can live inside an artificial culture media as well. Artificial culture media plus host, right? An example of this includes aspergillus and trachophytus are examples of facultative parasites, right? These are the examples of this trachophytus and aspergillus facultative parasites. And after that, when it comes to the other part, we are going to discuss about predators. Some of these also form predatory mode of nutrition. Predators in such a manner that some of these uh, you know predate nematodes. Example of this includes oyster mushroom, oyster mushroom, uh, Plutorus. Scientific name is Plutorus. Oyster mushroom, Plutorus. Scientific name is Plutorus, and what it actually does is that the it paralyzes the nematode. It paralyzes the nematode, and once it paralyzes the nematode, it tends to penetrate its hostoria within the nematode and absorb the nutrition, absorb the nitrogenous compounds from the nematode, as a result of which it tends to gain its nitrogenous compound requirement from the nematodes. And when it comes to its carbohydrate compound requirement, that is obtained from the cellulose present in the wood, present in its nearby environment. So nematodes are grown, they paralyze the nematodes and kill them. So as a result of this mode of nutrition in which they attack on they work on they obtain nutrition from herbivores, plants, as well as carnivores, animals. They would be considered as omnivores because they eat on feed on vegetative part as well as non-vegetative part, which means they feed on animals as well as plants. So these would be considered as omnivorous predators. Oyster mushroom is an omnivorous predator. After that, When it comes to another example, we are having Arthroboetrus. It is a genus. Arthrobotrys. It is a genus which basically causes to form a constriction ring around the nematodes, due to which that constriction ring tends to, you know, constrict the nematode and attach towards it to absorb the nutrients from the nematode, either intercellularly or intracellularly, such that when it penetrates into the nematode, it either intracellularly or intracellularly absorbs the nutritional material, such as the nitrogenous compounds primarily from the nematodes. After that, when it comes to another example, some other kinds also, perform, uh, release sticky substances out of their body, which help them to it entangle the nematode within their sticky secretions. Moreover, there are some other parts as well, such as, we are having some common examples of mushrooms and as well as puffballs, morels. These are those fungi which have, they develop their hyphae into reproductive structure, into a complete reproductive structure. Uh, um, they develop a complete, reproductive structure, um, a giant complete reproductive structure, which is either esco a complete giant reproductive structure, which is either known as ascocarp in ascomycotes if it is developed in ascomycetes, and basidiocarp if it is developed in the basidiomycetes, right? And as you can see in the mushroom, this structure is also developed in mushroom as well as in other types of fungi where there is development of this structure. As you can see in the diagram, these are the ascocarps and basidiocarps. Ascocarps, basidiocarps, when we'll come into classification, we'll further discuss that in detail. And these are some basic points. Hopefully, we'll meet again in another video.